Good morning and welcome to all. So today, after the completion of HMP shunt pathway, we are going to discuss about Entner Doudoroff pathway. The Entner Doudoroff pathway, it is the metabolic pathway that is most notably in gram-negative bacteria and also certain gram-positive bacteria. And they are also seen in certain archaebacteria also. Here, glucose is the starting product in the ED pathway. ED is the short form of Entner Doudoroff pathway. And through a series of enzyme-assisted chemical reactions, this pathway takes place. And finally, the glucose can be catabolized to pyruvate. Entner and Doudoroff in 1952 and McGee and Doudoroff in 1954 first reported the ED pathway in the bacterium Pseudomonas saccharophylla. Okay, while originally thought to be just an alternative to glycolysis, that is an Entner-Doudoroff pathway, it was thought it was an alternative to the glycolysis and also the pentose phosphate pathway. But some studies now suggest that original role of EMP, that is glycolysis, may have originally been about anabolism and repurposed over time to catabolism. Actually, we have studied that glycolysis is a catabolic process. But there are some studies that it was first originally about anabolic process, then it can be repurposed over time to catabolic process. And it means that this ED pathway also like that may be an older pathway. Now, what are the distinct features of the Entner Doudoroff pathway? It uses a unique enzyme, that is 6-phosphogluconate. 6-phosphogluconate dehydratase aldolase and 2-ketodeoxy 6-phosphogluconate aldolase and other common metabolic enzymes to other metabolic pathways. They are used to catabolize glucose to pyruvate. So the unique enzyme included is 6-phosphogluconate dehydratase aldolase, and there are two enzymes, 2-ketodeoxy 6-phosphogluconate. In the process of the breaking down of the glucose, there is a net yield of 1 ATP and it is formed per every 1 glucose molecule processed. So in this process, 1 ATP molecule is synthesized as well as 1 NADH and 1 NADPH. So 1 ATP, 1 NADH and 1 NADPH molecules are synthesized as a result of the process of breaking down of glucose. In comparison, the glycolysis has a net yield of 2 ATP molecules and 2 NADH molecule per every 1 glucose molecule we have glucose molecule metabolized. This we have already discussed that each glycolysis have net yield of 2 ATP molecule. Though the studies suggest this difference in energy production may be offset by the difference in the protein amount needed per pathway. So, the difference between the two cycles, that is glycolysis and this ED pathway, it is mainly due to the difference in the amount of protein which are needed per pathway. Now, next is the organisms that uses this pathway. There are several bacteria that uses this pathway for the metabolism of glucose and which are unable to catabolize through glycolysis. So, there are certain microorganisms which can't catabolize through glycolysis use this pathway. For example, the organisms who are lacking the essential glycolytic enzymes such as phosphofructokinase. It is an important enzyme in glycolysis. So certain bacteria such as Pseudomonas, they lack this enzyme. So they prefer alternative ED pathway than glycolysis. Okay. The genera in which the pathway is prominent include gram-negative as well as gram-positive bacteria such as Enterococcus faecalis. Enterococcus faecalis comes under gram-positive cocci, as well as several in the archaea, the second distinct branch of the prokaryotes. It is also present in the archaea. Archaea is the another branch. Originally, they have characters similar to prokaryotes, but they are not prokaryotes. So, they are coming in between the prokaryotes and eukaryotes. Due to the low energy yield of ED pathway, anaerobic bacteria seem to mainly use glycolysis, while aerobic and facultative anaerobes are more likely to have ED pathway. So, this ED pathway we are seeing more commonly in aerobic as well as facultative anaerobes, and they are less commonly seen in which anaerobes. Anaerobes usually prefer glycolysis. Thus, the ED pathway, it is favored due to the lesser amounts of protein required, while anaerobes must rely on glycolytic pathway to create greater percentage of the required ATP. Thus, its 2 ATP production is more favored over the ED's 1 ATP production. So, we have discussed that in ED pathway only 1 ATP molecule is synthesized, whereas in glycolysis 2 ATP molecules are produced. So, the anaerobes prefer glycolytic pathway because they require 2 ATP molecules, greater percentage of energy. Now, next is the examples of the bacteria using the pathway are Pseudomonas, a genus of gram-negative bacteria, Azotobacter, a genus of the genus of gram-negative bacteria, Rhizobium, a plant root associated and plant differentiation active genus of gram-negative bacteria. So, this Rhizobium is usually present in the soil. It is a plant root associated bacteria. Next is Agrobacterium. It is also a plant pathogen which are coming under gram-negative. They are also used in biotechnologic purpose. Then coming is Escherichia coli or E. coli. It is also a gram-negative bacteria. These are the organisms which utilize ED pathway. Enterococcus faecalis, a gram-positive bacterium. Zymomonas mobilis, a gram-negative facultative anaerobe. So, it is an anaerobe. Okay. Xanthomonas campestris. It is also a gram-negative bacterium which uses this pathway as the main pathway for providing energy. Xanthomonas campestris. Now coming to the catalyzing enzymes, how the process takes place. First of all, first step is the conversion of glucose to glucose 6-phosphate. The first step in the ED pathway, it is nothing but phosphorylation of the glucose by the family of certain enzymes called hexokinases to form glucose 6-phosphate. This reaction consumes ATP, but it acts to keep the glucose concentration low, promoting the continuous transport of glucose into the cell through plasma membrane transporters. In addition, it blocks the glucose from leaking out. And also the cell lacks the transporters for glucose 6-phosphate. So, the transporters are not present for this intermediate glucose 6-phosphate. This may result in the free diffusion out of the cell, and that can be prevented due to the charged nature of G6P. So, the charged nature prevents it from free diffusion because there is a chance of cell leaking out. So, because of the charged nature of G6P, the leaking can be prevented or diffusion can be prevented. The glucose may alternatively be formed from phosphorylases or hydrolysis of intracellular starch or glycogen. Next is the conversion of the glucose 6-phosphate to 6-phosphoglucanolactone. The G6P, it is then converted to 6-phosphoglucanolactone. Second step, in the presence of the enzyme glucose 6-phosphate dehydrogenase. And this is an oxidoreductase enzyme with the presence of a coenzyme NADP or nicotinamide adenine dinucleotide phosphate, NADP+. And this molecule NADP+ can be reduced to NADPH, that is nicotinamide adenine dinucleotide. Then, so NADPH along with the free hydrogen atom can be released as H+ ions. NADPH plus H+. Next is the conversion of 6-phosphoglucanolactone to 6-phosphogluconic acid. This step takes place in the presence of the enzyme hydrolase. This enzyme converts this 6-phosphoglucanolactone to 6-phosphogluconic acid. Next step is the conversion of 6-phosphogluconic acid to 2-keto-3-deoxy-6-phosphoglucanate. And this 6-phosphogluconic acid, this can be converted to 2-keto-3-deoxy-6-phosphogluconate KDPG in the presence of the enzyme 6-phosphogluconate dehydratase, in which the water molecule is released to the surroundings. So, this gluconic acid can be converted to gluconate, in which the water molecule can be released because of the enzyme dehydratase, which removes the water. Next is the conversion of 2-keto-3-deoxy-6-phosphoglucanate to pyruvate and glyceraldehyde 3-phosphate. So now this molecule converted either to pyruvate or into pyruvate or glyceraldehyde 3-phosphate with the help of the enzyme aldolase. This enzyme is converting KDBPG, that means 2-keto-3-deoxy-6-phosphoglucanate into pyruvate. The ED pathway for the pyruvate ends here, and then the pyruvate goes into further metabolic pathway. So now after the formation of this pyruvate, this may sometimes enter into either TCA cycle or electron transport chain cycle, etc. So the other product, that is glyceraldehyde 3-phosphate, this can be further converted by entering again into the, not again, it can enter into the glycolytic pathway, and at last it can be also converted to pyruvate for further metabolism. So two products, one is pyruvate and glyceraldehyde 3-phosphate. Pyruvate enter the TCA cycle or electron transport chain. Glyceraldehyde phosphate enters glycolysis. Next is the conversion of the glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. So this glyceraldehyde 3-phosphate converted to 1,3-bisphosphoglycerate in the presence of enzyme glyceraldehyde 3-phosphate dehydrogenase. It is an oxidoreductase enzyme. And the aldehyde group of the triose sugar, that is glyceraldehyde 3-phosphate, are oxidized and inorganic phosphate is added to them, forming 1,3-bisphosphoglycerate. The hydrogen is used to reduce two molecules of NAD+ and a hydrogen carrier to give NADH plus H+ for each triose. Hydrogen atom balance and charge balance are both maintained because phosphate group actually exists in the form of hydrogen phosphate anion, and this may contribute to extra H+ ion, and it gives a net charge of -3. So nothing but conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. This takes place in glycolytic pathway. Then conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate. This is the enzymatic transfer of phosphate group from 1,3-bisphosphoglycerate to ADP by phosphoglycerate kinase, forming ATP.